Electron microscopes enable biologists to see objects too small to see under a light microscope. Freeze fracture is a popular method for studying plasma membranes. Other organelles can be isolated and studied using cell fractionation. Large quantities of cells can be grown in culture when experiments require more cells than a tissue sample can provide. Let's take a look at each of these techniques to see what they can tell us about cell structure. Although light microscopes were invaluable at advancing our understanding of cells, they can't show objects smaller than about 0.2 micrometer. To distinguish objects smaller than this, biologists use an electron microscope, which creates the magnified images by using electrons instead of light waves. An electron gun emits a beam of high-energy electrons. This beam travels through lenses designed to focus the electrons on a very small spot. A detector counts the electrons emitted from each spot on the sample. This information is fed into a computer where the electron microscope reveals new levels of detail and complexity in the amazing world of microorganisms. Electron microscopes can distinguish biological objects as small as about 2 nanometers. This image was obtained using a scanning electron microscope, where the electrons bounce off the object's surface. Freeze fracture provides a way of visualizing plasma membranes. Cells are frozen in liquid nitrogen at -196 degrees Celsius. The frozen block is cracked with a special knife. The fracture plane usually passes through the hydrophobic interior of a membrane bilayer. The membrane faces are sprayed with a platinum-based compound to aid visualization. The original membrane is dissolved away, leaving a replica of the original membrane on a platinum film. The membrane replicas are viewed. When biologists are interested in viewing internal organelles, they use cell fractionation to isolate those structures. Tissues are homogenized and suspended in buffer solutions that help stabilize the cell's organelles. The mixture of plasma membrane fragments, cytosolic molecules, and unbroken organelles is placed in a centrifuge. This instrument spins samples at high speeds and separates components of a mixture on the basis of their size and density. At relatively slow speeds, only the largest, most dense particles form a pellet at the bottom of the tube. The first pellet usually contains the nuclei. A second, faster centrifugation of the supernatant, the liquid above the pellet, will cause the smaller, less dense organelles, such as mitochondria, to form a pellet. Extremely high speeds are needed to obtain pellets of individual intracellular structures like ribosomes, the protein synthesizing complex. Under certain conditions, most animal and plant cells can grow and divide in laboratory culture dishes. Cultures prepared directly from the tissues of an organ are called primary cultures. Cells from certain primary cultures can also be divided into multiple secondary cultures. This process can sometimes be repeated for weeks, months, or even longer. Cultured cells are often said to be growing in vitro, which literally means in glass. Cultured cells can often reduce the need for in vivo experiments on living organisms. Cultured cells are often frozen